All right. Good afternoon, everybody. I hope everybody's day is going well. We're gonna go through auditory, and then we'll go through olfaction, as well as taste. Right. We'll talk a little bit about chemesthesis at the end of it. But like I told the morning lecture or the morning session, I told them that I posted the PowerPoint already. Right. So the PowerPoint's online just because I made a lot of edits in regards to specifically the olfaction lecture. So I wanted to make sure you guys can tag along and basically follow the notes. Those two are posted, so feel free to grab them and take notes from there, and then we will go on with the session. So does anybody have any questions regarding anything, regarding neuro, program, etc. before we start? All right. If not, let me just pull up the PowerPoints and we'll get ready. All right. So we're going to be talking about audition. So we'll start with that. Let me know if I'm talking too fast, if you want me to slow down, or if you have any questions. Again, let me know. It's very hard when I'm talking to a screen how my pace is, right? So it'd be much easier if this was in person. But again, no shame. Just let me know if you want me to slow down or explain something again. I got you, okay? So we will go through audition first. So here are the objectives, right? So we'll talk about those in great detail. So the first thing we want to understand is what is sound, right? So what we listen to has four major features, right? We have waveform, phase, amplitude, and frequency, right? So those three different things are going to be things are very important for how we hear things. So amplitude is going to be loudness, also known as volume, and then frequency is going to be pitch, right? So that it's going to be very important that we define this now so that we don't get confused later on when we say amplitude. We're talking about how loud the sound is. When we talk about frequency, we're going to talk about the pitch basically of the sound. So here she talks a little bit just about simple sounds. So she wanted to start off about talking with she wanted to start off about this lecture basically with a tuning fork, which is going to do simple sounds. So it does the most simple form of sound imaginable. We don't listen in simple sounds, but this is just a concept for us to understand how sound works. So basically what happens is that with the tuning fork, if you were to bang a tuning fork on a desk, for example, it'd vibrate, right? And it makes a noise. How does it make this noise? So it basically makes this sound by differences of compression and rarefactions. And what that is basically when I'll show you on the next slide. I think it's easier to say that. So I'll show you in the slide. Okay, so basically what's happening is that when you hit that tuning fork, basically, the tuning fork is then going to open up as shown as like number 4. When that happens, basically, the tuning fork is going to compress the air right next to it, right? So it's compressing those air molecules. And then when it gets back to 2, like this, so it goes back together because it's vibrating back and forth, it's going to rarefact or rarefraction or rarefy. And when that happens, basically the molecules are going to come back into place, right? So molecules are just compressing, going back to place, compressing, going back to place. And that's how that sound is basically being transmitted across the air medium, right? So it's very cool, just a super simple sound wave in regards to a tuning fork, right? So compressions and rarefactions. And that's exactly what's shown here, right? So the right prong, for example, is going to be hit against the table. Right? When that happens, air right next to it is going to be compressed. Then air is going to be rarefied or, rarefaction. And then it's basically going back to place. And then it's going to basically keep going down a line. And what's happening is that this air, these molecules are moving along their own radius, right? So the molecule is not, say when you hit this tuning fork, one molecule is not flying like a mile away, right? The molecules stay relatively in their own radiuses, right? So they move, but then they come back to place. They move a little, they come back into place. So they have their own radius, right? The molecules aren't just flying across the air medium. All right, so that's basically what's happening here in regards to compressions and rarefactions, just like what we said, molecules coming together and then separating. Right. So again, like I just stated before, remember molecules are basically moving in their own medium. right? You can see in this picture, you can see the molecules basically moving side to side, eventually going back into their own original spot, right? So they kind of just like have their own little radiuses of compressing and then rareifying, but they're not going to be just transferring down the entire medium, right? That's not how the particles move. So as you can see, that's basically being shown in this picture on the right. So again, when we think about compressions and rarefactions, basically we have to think about when the molecules come close, it's compression. When they go away, it's or go back to normal, it's rarefaction or rarefaction. I can't say it, so hard. So basically, they thought about making this into a waveform. It's just a series of compressions and rarefactions. So basically, when it goes up, compression, down, rarefaction or rarefaction, it's basically they just put it into a waveform, right? So it's exactly what we talked about, but they just showed it in a wave, right? So, and this is when we're going to talk about the types of sounds and frequencies. So that's exactly what we talked about in the entire slide, right? Or the upcoming slides that we just talked about. So sinusoidal waves, what that is basically is showing that tuning fork basically with its compressions and its rarefactions, right? So going up, going down, going up, and then going down, right? That's basically of a perfect sound, right? So just going up, down, up, down. So they want to show that in a wave because it's much easier to see it on a graph like this, right? So when we talk about basically these waves, basically of these molecules just compressing and refracting, again, these molecules are going within their own radius. So you can kind of think of it as like a circle, right? So the molecule can move left, right, back, forth, etc. But basically at the end of the day, the molecule is just moving around in a small area and it's coming back to normal. That's where they got the idea of this basically phase angle. So what's happening basically is that these molecules are moving within their own radius, right? So you can see that's why we form this phase angle. And the faster the molecule can move around their own radius, the faster the frequency, right? Which makes a lot of sense because if the molecule moves a lot in its radius super quickly, just think about the molecule going like this super quickly, it has a fast frequency, right? And we indicate that or we depict that in a waveform when we draw it on a graph, right? The frequencies are very fast, right? So the waves look very fast. But here they just depict it as a circle, saying if the radius spins around the circle, the faster it spins around the circle, basically means the higher the frequency, right? And that's how this sinusoidal function basically associates with that waveform. So you can see in this image right here, you can see in A all the way until H, the faster this R basically or radius wraps around the circle, right, the faster the frequency, right? And that's basically what's going on here when it comes to molecules moving around their own radius. Okay, so phase. What's important about phase is basically that things or waves basically that have the same frequency and displaced in the same time are going to be in phase. So for example, if this wave right here, this one right here, match this wave right here, basically right on top of it, they would be in phase. The reason being is that the waves are going to have the same exact frequency, right? So if they have the same exact frequency and they're basically on top of each other, they're in phase. These two waves right here have the same frequency, but you can tell they're not in phase, right? Because this one started a little later than the other one. So they're technically 45 degrees out of phase, right? So frequency is what determines if something is in phase or out of phase. As you can see, this one's 90 degrees out of phase, and this one's going to be 180 degrees, which is basically the complete opposite, right? And that's basically the hearing of a simple sound, right? But obviously, human ears are going to hear complex sounds, things that are not pure tones or simple harmonic tones. So here, this is a great example for us to understand the difference between frequency and amplitude. So you can see this picture on the left and this picture on the right, they have the same amplitude, right? Because you see the height of the wave is exactly the same on the left image than it is on the right image. The difference is that their frequencies are much different. This one is much slower than this one, right? Hence why this is 100 hertz and this is 500 hertz, right? So this means both sounds are just as loud, right? So they're both just as loud, but this one basically has a higher frequency, while this one has a lower frequency, right? So good example, like birds chirping is an example of a very high frequency, right? While something low frequency, I would say, is more so like a tuba, right? Like a tuba or a bass. Those types of instruments have very low frequencies. They can be just as loud, right? Both of them can be just as loud, but the bird chirping is going to have a higher frequency. You see what I'm saying? So those are the big differences between frequency and amplitude. So we hear basically best between 20 hertz and 20,000 hertz. We hear between 20 hertz and 20,000 hertz. So that's important to note, right? We want to know that we can hear between that range, and that's interesting because the male speech and female speech is perfectly in line within that range, and that's not a coincidence because, I mean, communication is very important for humans, right? So it would make sense that male speech and female speech would be in the middle of our perfect hearing range, right? We want to be able to hear each other and communicate with each other, right? So over here, regarding this graph, basically we are more sensitive to certain frequencies than others. So yes, we can hear from 20 hertz to 20,000 hertz, but we're best in the range of 2,000 to 5,000. And that's shown in this graph, right? So if you look at this red box right here, around 2,000 to 5,000 hertz, all you need to do is hear the sound at roughly around 10 decibels, right? Which is very, very low, right? That's lower than a whisper. If something like, for example, if something has a frequency or a sound has a frequency between 2,000 and 5,000, all you need to do is hear it at like a whisper loudness, basically, to hear it. But if something had, for example, like a 10 frequency, you would need it to be almost 120 decibels in order to hear that, right? So you can see how this 2,000 to 5,000 hertz, we're much more sensitive because we can hear at a much lower threshold. This yellow line is kind of like our uncomfortable loudness line. Once you're above 100, basically, your ears kind of feel very uncomfortable, right? But when you're in this red range, you can feel a lot of pain and can lead to damage. So in summary, basically, the best frequencies, we hear at the best frequencies that are most important to us, right? So humans, communication is very important to us, so we hear best at communication frequencies or human communication frequencies. Dolphins, they have very, very high frequencies way out of our range that are important for their communications, right? And then over here, chickens are going to hear at very low frequencies. Okay, so if two sound waves are in phase, they must have the same blank. Any questions? It's a little trick question. I'll give you a second. Okay, so a question? I think somebody's mic's on. Okay, so if two sound waves are in phase, they must have the same frequency, right? So it was kind of a trick question, right? So it could have, I see how you guys think it was more than one answer. That's the reason why I put this here. So you don't have to have the same starting time to be in phase, right? If the same frequency was to start on the second wave, basically, or the third wave or the fourth wave, they'll still be in phase because the frequencies are exactly the same, right? So yes, starting time is great, right? If it starts at the same time and has the same frequency, it will be in phase, but it doesn't have to start at the same time. That's why the only thing that must happen is that they must have the same frequency in order to be in phase. Starting time is nice, but it's not a must, right? Something can start way later and still end up being in phase, right? So I'd say the answer is B. I think somebody's mic is on. Okay, so we have peripheral to central. So we'll talk about hearing. So external ear, you have your pinna, your concha, and your external auditory meatus, right? So these are the three things you're going to want to know in regards to hearing, basically. The pinna we'll talk a lot about in the next slide, but basically the pinna is very important, like you saw in the quiz, in elevation and depression localization. So if a bird is chirping on the ceiling, basically, or when whatever, top of the building, how do you know that it's coming from the top? Your pinna is very, very important in that. And they did a study basically on that. Um, I think it's the slide after that, in which they used an artificial pinna in order to track basically if the pinna was important for sound localization, and it is. Specifically, it's more so associated with elevation and detection, or elevation and depression, right? But again, we know that the external ear is important for protection, right? It protects, for example, um, insects, etc., bugs, whatever, from getting into the ear. It also gathers a frequency boost of 3K hertz, and then again, sound localization, like we just talked about. And this is the study we just stated. Remember, monaural localization is basically one ear. So basically, it just shows that the pinna is important in detecting sound from one side of the ear, or detecting the localization of that sound on one side of the ear. Okay, so again, that's just your outer ear. Again, just note the three different things: concha, pinna, and external auditory meatus, and that the pinna is very important in sound localization. Okay, so tympanic membrane is going to separate the outer and the middle ear, right? So our middle ear is basically going to contain our malleus, incus, and stapes. The way I like to remember that is MIS, so miss, malleus, incus, stapes. And this is the cool thing that she talked about. So basically, the thing that you want to know is that the stapes is going to stomp on the oval window, right? You want to know that it's the oval window, not the round window, because I know that kind of gets confusing, but it's the oval window that the stapes is going to stomp on in order to allow for transmission of sound. Okay, so in regards to the middle ear is basically going to do impedance matching, and what that is you have to think that the air that we listen to is obviously the sounds through air are through an air medium. The inner ear is actually going to be a fluid medium. So you have to think that there's going to be an impedance mismatching between the air and the fluid. A great example is shown here, like she showed us. If you call somebody's name outside of the water and they're swimming in the water, it's going to be very hard for them to hear you because of this impedance mismatch basically between air and water. But if someone was to click two rocks together in water, they would hear it much, much better than you screaming from the top, basically outside of the water. You have to think about that basically in the same way as how the ear works. That middle ear is basically trying to make sure that we don't lose any decibels going from an air medium to a fluid medium, right? And the middle ear is very, very important in that. So how does it do that basically? So the middle ear does that via three mechanisms. It has area advantage. So the best way to think about that is you have a thumbtack, right? And a thumbtack basically on the back side, it's a very, very large area. And on the pin side, it's very, very small, right? So when you put it into the wall, you put a lot of pressure on the back side, which is a larger area. And then that same pressure is going to be exerted on that super, super small pin, right? But it's going to be a much smaller area. So that's how we get that area advantage here. The tympanic membrane is much, much larger, and that same force from the tympanic membrane is going onto this much, much smaller stapes window, right? So that helps us preserve some sound. It also has an acicular lever action. So basically, when the tympanic membrane is cranked one way, it cranks the other way via the stapes and allows for another 1.3 decibels. And lastly, you have a curved membrane in the tympanic membrane. The tympanic membrane is literally curved, allowing for sound basically to hit the most middle part of the tympanic membrane to allow for additional 6 decibels. Right? So that's how those are the three mechanisms in which the middle ear is going to lead to that impedance matching. Okay. So you have your inner ear. So now we're in the inner ear. You know that your inner ear has your cochlea as well as your semicircular canals. Your semicircular canals we know are important in vestibular senses, right? So for balance. And we know that your cochlea is important for hearing, right? So we're going to talk about the organ of Corti, which is located in the scala media, which has endolymph, and scala vestibuli and scala tympani have paralymph, and they have much, much different functions in regards to their fluid. I think somebody's mic's on. Is anybody? You could turn your mic off, please. Okay, so hold on, let me see. Whose mic is it? Okay, got it. Okay. Just fix that. Okay, so now we have our inner ear. Basically, the cochlea is as a traveling wave, right? So basically, as the stapes goes in and out, basically of that oval window, it creates a wave in the cochlea, right? Remember, again, the stapes is pounding on that oval window, and then it's moving that scalar media up, down, etc. Right? It's going to be very important in how we transmit sound. So here, when that stapes pounds on that oval window, it's basically pushing this fluid called perilymph, right? And it's pushing perilymph all the way around the cochlea via the scala vestibuli first, right? So this top portion first, which is called the scala vestibuli, going all the way up, all the way to the helicotrema, when it then turns back down via the scala tympani, right? So the perilymph does not go into the scala media. The scala media has endolymph, right? So that makes sense because when the stapes window is pounding on into that scala vestibuli, perilymph is moving around the entire cochlea. This causes basically vibrations of that scala media, which we'll talk about how important that is later on. But again, perilymph is not going to have any electrical charge, so it basically doesn't, it's not important for sound hearing, right? Because it doesn't have any electrical charge. Endolymph actually has a positive 80 millivolts, and that's actually going to be very important for conducting sound signals in the scala media due to an electrochemical gradient that will help cause action potentials, right? So here basically she talked a little bit about how the scala tympani, the division of the scala tympani and scala media is going to be that basilar membrane. We're going to talk a lot about the basilar membrane, so that's going to be super important. The barrier between the scala media and the scala vestibuli will be the Reisner's membrane, and I think this is the only time in the lecture we'll talk about that, so not too high yield in regards to that. So tonotopic organization of the basilar membrane. So to no surprise, we have tonotoxicity almost everywhere, right? So even in the basilar membrane. So depending on the frequency is going to, that sound is going to basically signal a receptor in a certain location, right? So for example, the highest frequencies are going to be going to the base, right? So they're going to the base, which is going to be the most stiff part. The lowest frequencies are going to be going to the apex, which is the least stiff part, right? So the way I like to remember that is all, so basically ALL, apex is going to be least stiff, and it's going to be lowest frequencies, right? So the base must be the opposite. That's the way I remember it. So again, apex is going to be, or all is the way I remember it, apex, least stiff, lowest frequencies. Base is just the opposite, right? So you can see basically that's what I'm talking about here. Here's our apex, least stiff, lowest frequencies. In our base, we have most stiff and then highest frequencies. You can see if you stimulate certain frequencies, it's going to tune in different areas of the cochlea, right? So this shows how we have tonotoxicity in the cochlea. So here, this is basically the organ of Corti. It's going to be a complex structure composing of hair cells and other cells, but we're going to zoom in into this red box right here. But again, note that the scala media is going to have endolymph, right, which is very important for helping with action potentials within the scala media. Okay, so the organ of Corti basically is going to have 3 rows of outer hair cells and 1 row of inner hair cells, right? So we have much more outer hair cells than we do inner hair cells. And remember that this basilar membrane is going to be that separation between the scala tympani and the scala media, right? So this entire thing right here is the scala media. Down here is going to be the scala tympani, right? So here's our tectorial membrane. And then we're going to talk a lot about the outer hair cells So this point on the bottom is going to be really important later on, but I would just start for now because we're definitely going to know that later on, but I'll explain it when we get to that slide. So if your neighbor is playing the tuba, which is a low frequency, which part of the cochlea would that sound most likely hit? Okay, any questions? Okay, so if your neighbor is playing the tuba, which is a low frequency, which part of the cochlea would that sound most likely hit? And we know low frequency, remember that's going to be associated with the apex. So that's our all, apex, least stiff, lowest frequency. Bass is just the opposite. Right? So when we think about this, we're like, okay, activation of hair cells, we know it's hitting that tympanic membrane. Then it's going to hit that stapes foot plate. So it's going in and out. Basilar membranes moving up and down, but how are these cells activated, right? How do we activate these cells and transmit the signal, right? So we have excitation of hair cells, right? So how does this happen? So basically what's happening is that basilar membrane that we showed in the last picture is moving upward. When that basilar membrane moves upward, it causes the hair cells, the cilia of the hair cells, to bend laterally, right? So when they bend laterally, they open their pores and basically lead to excitation of the hair cells, right? So I'll show you that on the next slide. It'll make a lot more sense. So here you can see in this top portion, you can see the basilar membrane is moving upward, right? So it's moving upward, and then these hair cells right here, you can see there's lateral bending here due to that shear force, right? So you see the lateral bending, and that's opening up channels right up here, causing neural signals to be transmitted, right? So the way you could think about it, I mean, you could think up, right? So up and lateral, up and away, like things like that can help you remember that that's going to be activation or excitation. Inhibition is actually going to be when the basilar membrane moves down and the cells go medial, right? So down and medial, whichever way you want to think about it. But down and medial are going to be inhibition, right? So it's going to inhibit basically the channels, right? Because you can see the channels were closed in this angle, but they would open here, right? So reason being is because we have these tip link structures, right? So you can see in the stereocilia, these tip link structures are connecting basically different potassium channels, right? So when these hair cells move up and lateral, right? So up and away or up and lateral, that's going to be excitation, right? Excitation, potassium goes into the cell, depolarization, and calcium goes into the cell, leading to further depolarization. This is important, right? Because it's kind of weird because we're not used to potassium going inside of the cell, but potassium going inside of the cell is causing a depolarization because that's more positive charges going inside of the cell. And then eventually leading to an action potential, right? And this is going to be basically that mechanoelectric transducer, right? So this a mechanoelectric transducer, what's happening is that, remember, that basilar membrane that moves up and then the stereocilia bend lateral, those are going to open up those tip link structures. So you can see these tip link structures are opening up channels, right? And eventually, obviously, potassium goes in. And we know that actin and myosin are going to play crucial roles because those are motor proteins. They help with movement, right? So that's going to be very important as well. So inhibition, again, if up and lateral is excitation, then down and medial is going to be inhibition, right? So when the basilar membrane goes down and then the hair cells go medial, that is going to be inhibition, right? So it's very, very important for us to know the differences between those two. So once that happens, we're like, okay, what about the hair cells themselves? Well, so the hair cells themselves, we have outer hair cells versus inner hair cells, right? We have a lot more outer hair cells, and way less inner hair cells. And what is the difference between their function? So basically, outer hair cells are basically the life of the party, right? So in the party, basically they bounce up and down when they hear sounds. You could, when she showed the video, you saw the outer hair cell bouncing up and down, and you know that the outer hair cell is very important in that amplification of sound, right? So amplification of sound is outer hair cell. Inner hair cell is super important in neural signal transduction, basically turning that mechanical signal of the outer hair cells basically going up and down into a neural signal into the auditory nerve, right? So here basically outer hair cells, like we said, they're going to bounce up and down when they hear noises, right? And what's happening is that they go through excitation and inhibition. So excitation, the cell is going to shorten. It's very important for us to know, this is super important, definitely going to get tested on this, is that the Preston protein is going to be important for the outer hair cells, right? So it's going to help, it's going to be important for outer hair cell motility, right? So think about it. When the basilar membrane moves upward, it's going to push those hair cells like onto the tectorial membrane. Thus, we know when the basilar membrane moves up, the cilia, the stereocilia move lateral, and the cells are kind of getting, crunched or a little shorter. That's when we know, oh, excitation, the outer hair cells are getting shorter. When the basilar membrane moves downward, we know that the cells are elongating, right? So they're getting longer, and that's going to lead to inhibition. And then remember that the stereocilia move medial, right? So that's very important for us to know those differences. So exactly what we talked about, upward versus downward movement of the basilar membrane leading to excitation or inhibition. And remember, the Preston protein is very, very important in the motility of outer hair cells. Okay. Again, remember, outer hair cells amplify sound. That's their main job, the amplification of sound. Inner hair cells, remember, those are going to be super important in transducing signals from mechanical to neuro, right? So that's exactly what we see on the next slide. Inner hair cells are important of transducing mechanical vibrations into neural signals. So if the basilar membrane were to move upward, where would the hair cells bend and what event would that lead to? Give you a few more seconds. Does anybody have any questions? Everything seems clear then? Okay. So I see the answers in the chat. The chat looks good. If the basilar membrane were to move upward, where would the hair cells bend? Remember, upward in a way, so upward and lateral. So that's how the hair cells are going to bend, right? And lateral bending leads to excitation, right? So lateral bending, excitation. Remember, medial bending is inhibition, and the basilar membrane will move downward. Okay. So activation of hair cells, again, we have our sound coming into our tympanic membrane, leading to vibrations, Stapes foot plate going in and out of that oval window. Basilar membrane moves up or down, depending if it's excitation or inhibition. And then hair cells are activating those auditory nerve fibers. It's really interesting because this all happens within nanoseconds, right? You listening to me right now, all that sound basically going into your ear is within nanoseconds. That's how quickly this, like we're going through like, an hour of talking about the ear, and the ear is working in nanoseconds, right? So it's just kind of crazy how fast the ear works in order to pick up sounds. And not only pick them up, like understand them too. So we have receptor potential. She glanced over this slide a little bit, but she barely talked about it. But the differences basically between temporal coding. Temporal coding is more so associated with lower frequencies. It's kind of like each time a sound basically hits your ear, it generates basically a certain action potential at that same time into the auditory nerve, right? So basically leading to consistent firing at each sound point, basically. When you're at higher frequencies, it's much harder to do that, right? Because everything is super fast. So instead, we rely more like on this labeled line coding, which means that you have more so of that topographic distribution, right? The frequencies, depending on the frequency, is going to depend on where it lands on the cochlea. Yeah, but this is very low. Like, she didn't talk about this too much, but those are the main differences between those two. A little bit that she adds onto that is this right here when she talks about the tonotoxicity, right, which is like or labeled line coding within the auditory nerve fibers. So we said, remember, the cochlea has auditory or has tonotoxicity, right? And what that is basically is going to be, again, certain frequencies are going to be activated at certain spots, right? And there's a 1-to-1 ratio here between inner hair cells and the eighth nerve fibers, right? Which is really interesting because we know we have, I think, I don't know, like 1,500 inner hair cells, meaning that we must have around 1,500 axons of the auditory eighth nerve, right, in order to match those inner hair cells and eventually form that auditory nerve. Right, so we know that's going to be very important, and this tonotoxicity is going to be related to the characteristic frequency, which just means the frequency the hair cell best responds to. So if a hair cell is like, I want to respond to 100 hertz, then the auditory nerve fiber that's associated with that inner hair cell is like, okay, then I guess I have to respond to 100 hertz, right? So this means that there's tonotoxicity within the hair cells as well as within the auditory nerve fibers. And again, that's just what we talked about here. Just that characteristic frequency, which is the frequency of the greatest sensitivity for that fiber. Okay, so you have type 2 fibers versus type 1 fibers. The way I like to think about it is type 2 fibers bind to too many. So basically it binds to multiple outer hair cells. Type 1 fibers, it's 1-to-1. So type 1 fibers bind 1-to-1 to inner hair cells, right? And eventually, obviously, these are going to form that auditory nerve or the cochlear portion of that auditory nerve. So central auditory pathways. Does anybody have any questions before we talk about this? Okay, I hope everything's crystal clear and everything like that. You guys are good. Okay, so here basically you want to know this pathway for sure. She will definitely test on it. And specifically, you want to know, I remember this being important last year, you want to know the lateral lemniscus. It's not on this pathway shown here, but it was shown in the video, right? And hint, I remember that being very important, but because it wasn't written here. So, but you want to know that the auditory nerve, yes, it goes to the cochlear nucleus, then the SOC, but from the SOC to the inferior colliculus, there's this thing called the lateral lemniscus, right? You're going to want to know that. Then it goes to inferior colliculus, thalamus, the medial geniculate body, and the auditory cortex, right? So you're going to want to note this pathway. Listen to the video a few times. I did. It helps you remember every single thing within that pathway, but specifically, you definitely want to know that lateral lemniscus. Okay, so cochlear nucleus, that's basically going to be the first auditory nucleus, right? So sound comes into the ear, eventually goes through all these processes, goes into that auditory nerve, and then into the cochlear nucleus, right? The cochlear nucleus is actually the only pathway centrally that's going to be monaural, basically, so only get one ear. Everything after this gets signals from both ears, right? So the cochlear nucleus, for example, on the right side only gets the right ear, on the left side only gets the left ear, right? So of course it's going to relay signals to the superior olivary complex, even the inferior colliculus, but again, it's going to be monaural, right, which is important. So now we have our SOC, or superior olivary complex. It's located within the pons. It's both ears, and very important for sound localization, right? So we have our lateral superior olive, right, versus our medial superior olive. Lateral superior olive is high frequencies and interoral level differences, right? Medial superior olive is low frequencies, interoral time differences. One way I like to remember this is lateral superior olive, L for level, so it's interoral level differences. Medial would be interoral time differences, right? And I'm going to explain what that means in the next upcoming slides, and it'll make a lot more sense, I promise. And again, to no surprise, we have tonotoxicity in the lateral superior olive as well. So this basically what's going on here. Again, remember that lateral superior olive is going to be that interoral level difference, right? So for example, remember the lateral superior olive is going to be at higher frequencies, right? So if you were to play basically a speaker from one ear basically to the other, right? Let's just say you're playing a speaker on your right ear, right? So you hear a sound from your right ear. How do you know that sound is going from your right ear? Okay, let's say this sound you said has a very high frequency because that's what the lateral superior olive is important for. Things that have very high frequencies can get around the head and into the left ear very, very quickly, right? Because the waves are super fast, right? So it hits the right ear and hits the left ear very, very quickly. So how do we tell the difference between the right and left ear with high frequencies? Well, we tell the difference between their level, between their loudness. Which one is louder? If it's louder on the right ear, we know it's coming from the right side. If it's louder on the left ear, we know it's coming from the left side. Right? So that's how we can tell the differences between things that are at very high frequencies is via their level difference, their loudness difference. Right? You go through this pathway inhibiting the right side if it's heard on the left or vice versa. And eventually this is going to tell us basically the differences between where the sound is coming from when things are at high frequencies. Right? So that's with high frequencies. With the medial superior olive, that's going to be at low frequencies, and that's going to be reliant on interoral time differences, right? And I'll explain what that means. So interoral time differences, remember, so now we're talking about low frequencies. With things that have very like slow waves, right? It takes a long time for it to go from the right ear to the left ear, right? The frequencies aren't as fast. So then things that are at low frequencies, we can just rely on the time difference between it hits the right ear and the left ear. Remember when we talked about high frequencies, because they're so fast, by the time it hits the right and left ear, time difference is almost negligible. So how are we going to be able to tell the difference between if the sound is coming from the right or the sound is coming from the left at a high frequency? Well, we dictate it on level difference, right? How loud it is, right? That's why we use that for the lateral superior olive. The medial superior olive, we know, okay, these are low frequencies, right? So we can use this to our advantage because low frequencies are going to hit one ear way faster than the other. So we can use that time difference basically in low frequencies in order to dictate, is this coming from the right side or is this sound coming from the left side, right? So now that's when this medial superior olive comes in because now we're talking about lower frequencies, so we rely on that interoral time difference in order to understand where these sounds are coming from. So here, basically, you can see interoral time difference. That's going to be things that are going to have lower frequencies, right? Because lower frequencies, it takes a longer time to get from the right ear to the left ear, so we can rely on that with low frequencies. Things that have high frequencies, because the time it goes from the right ear to the left ear is almost negligible, we need to use the level difference, which is going to be the difference in sound, basically, right? So how loud something is. So if you wanted to know which side of you a bird was chirping, high frequency, at which area would be activated, or which area would be activated, and what is correctly associated with that area. Any questions? Does that make sense? I know it was kind of confusing when I saw it last year, so I hope that clears things up. Okay. Seems like everybody's on the same page. So what's going on here is, remember, if this is a high frequency, right? So if it's high frequency, you have to tell yourself, okay, by the time it hits the right ear versus the time it hits the left ear, it's going to be negligible. So it can't be interoral time difference. We're going to rely on that interoral level difference, right? For that. And that's going to be associated with the lateral superior olive, right? So lateral superior olive is higher frequencies. medial superior olive is lower frequencies. Okay, so now we have our inferior colliculus. We know it's going to be important for auditory reflexes, so like our head turn response, as well as even responses with some visual cues. Here we have our medial geniculate body. What's very important about this is that, yes, we know the fibers, the sounds are going to travel that cochlear nucleus, superior olivary complex, et cetera, but some, can skip some portions of the pathway, right? They can jump some of the portions, but it will never jump the medial geniculate body of the thalamus. Every sound basically has to go to the thalamus in order to get to the cortex, right? So that's going to be that gateway for that. So this is a little summary. So almost all the pathways are crossed, so they're all binaural, except the cochlear nucleus. That's going to be monaural. You have tonal toxicity, as well as spatial localization and feedback. So we'll talk a lot about the feedback as well. So the auditory cortex, so after we talk about auditory cortex, we know has tonal toxicity, right? So once the sounds get in there, eventually sends it to the secondary auditory cortex and other association areas to determine, like, do we like the sound? What do we need to respond to the sound, et cetera, et cetera. So shown here, you can see the 500 hertz all the way up to 1600 hertz, 16,000 hertz. You can see that the auditory cortex even has tonal toxicity as well. So in this experiment, it basically just told us that sound is very preserved, right? So if you were to play something into your ear and then play the waveforms basically from the brain, it's basically going to be almost like playing the exact same sound. Basically saying that sound is very preserved throughout the entire process of auditory function, which is quite phenomenal. So auditory cortex, we know that's auditory memory, discriminations, as well as non-reflexive motor responses. Remember, non-reflexive, because reflexive motor responses is more so associated with the inferior colliculus. Okay, and we're going to talk a little bit about the efferent system, and we'll end off here for audition. But this is basically going to be a way in us to control the sound that is coming in, right? Of course, we have a feedback mechanism, which would be efferent, telling basically the hair cells, do we need to listen to the sound or amplify this sound, etc. Efferent innervation, right? We have roughly around 1,600 efferent auditory nerve fibers, and one of the ones is going to be the olivocochlear bundle, right? This is coming from the superior olivary complex into the cochlea. Contacts outer hair cells or inner hair cells, depending on the olivocochlear bundle, which we'll talk about. But again, remember, what's important about this is that this is like important when you're sitting in a restaurant, right? And you're trying to talk to somebody and you hear obviously a lot of noise because the restaurant's very loud. Usually, block off that noise, but how do you block off that noise and focus on who you're talking to? It's this efferent pathway, right? Telling the hair cells, hey, don't pay attention to the background noise, right? So that's exactly what these cells are important for. So we have two pathways that we need to know. That's why I put a star here. You have your uncrossed olivocochlear bundle and your crossed. Uncrossed is going from your lateral superior olive to your inner hair cells. Your crossed is basically going from your medial superior olive into your outer hair cells. So the way I like to remember that is that the crossed olivocochlear bundle, so you cross the street outside, right? So that makes me remember, okay, the crossed bundle is outside, so outer hair cells, right? So the crossed olivocochlear bundle is going to be for the outer hair cells, while the uncrossed is going to be for the inner hair cells. Okay, and that's exactly what's shown here. You can see the crossed olivocochlear bundle from the medial superior olive, right, is going to come and cross this side. The lateral superior olive is going to stay on the same side and stay uncrossed, but go to the inner hair cells. And again, that's what that pathway is important for. It's important for trying to basically block off noise in a loud restaurant. That's why these efferent pathways are super, super important. Right, so again, the ones that are going to bind to the outer hair cells are going to dampen it, right? So we know that the crossed one, because you cross the street outside, so the crossed olivocochlear bundle is important for dampening that sound because it's innervating the outer hair cells, which are the amplifiers of sound. so you can tell the amplifiers of sound to dampen it, don't pay attention to it, right? Or it can regulate the inner hair cells as well. And that is it in regards to this lecture. Anything after this is not going to be tested on. She said no clinical questions for the exam. Cool. Any questions?
Justin, I have a question. So is there anything that isn't tonotopically represented, or is it everything, like all nerves and stuff?
Yeah, I mean it's almost all of it. I think we'll talk about olfaction when there's some things that aren't tonotopic, but most of it's gonna be.
Okay, thank you.
Yeah, no problem. Okay. So we'll take like a 2 minute break or 2 3 minutes. So we'll start like 5:54, 5:55, and then we'll get right to it, and then we'll finish this last lecture of olfaction. All right, so we'll get to it. Make sure you guys have the olfaction lecture that's posted online. We'll be referring to that. So I made a lot of notes there, so we'll be going through that lecture. Let me just share my screen. And then we should be ready to go. This lecture I know is really important. I remember a lot of questions being on this from the exam, and this is like one of the toughest lectures I feel like for me. Just understanding the concept was quite difficult, but make sure if you guys have any questions, let me know, and I'll try and do my best to explain it as digestible as possible. Okay, so we have olfaction and taste. So here are the learning objectives. So basically, our smell is fascinating. We can basically discriminate greater than 1 trillion stimuli, right? So we can discriminate a lot of different smells, right? And this is the pathway you can see in this image right here. When something, let's just say you smell something, what's the pathway that goes on? So the smell comes into your nasal cavity, eventually binding to these cilia, which have receptors, right? Going to the olfactory receptor, through the cribriform plate, synapsing at this glomerulus, and eventually through the mitral cells and tufted cells, it goes into the olfactory bulb, right? So that's the general pathway of olfactory system. Excuse me. But there's no synapses within the olfactory epithelium, right? So this is the olfactory epithelium right here. There's no synapses right there, right? So that's important for us to note. So olfactory receptor neurons, they're bipolar, right? They have dendrites forming, cilia and axons like we just showed in the last image. Here in B, you can see the cilia. And then basically, again, no synapses within the olfactory epithelium. So, and you can see these axon bundles right here, which is shown in AX. Okay, nothing he'll test on here. But what's really cool is that you can see the difference between almond and urine is very, very slight. I believe this is an aldehyde and this is a carboxylic acid. Very, very similar right here, right? So, and you can tell that there's a big difference in the smell of urine and almond, but there's very slight difference molecularly. So it's super interesting, or super cool how the smell or a sense of smell can discriminate smells this close alike in regards to their molecular structure. So odor receptors are just going to be the largest family of receptors in the entire genome, right? So we have, I think it's like close to 1,000 odor receptors. I think they show us on the next slide. Yeah, close to 1,000 odor receptors and roughly around 400 to 500 genes. So we have more receptors than genes, which makes sense because we have splicing, right? So splicing can make a more variance. Right, and all the odor receptors are going to be GPCR, right? Important for you to note that these are going to be GPCRs. We should be familiar with GPCRs. We talked about them throughout the entire block. Basically, remember, those are going to be going through that 7 transmembrane domain located right here, right? So here, again, to no surprise, olfactory epithelium is having a topographic distribution. I think that should be good. Okay, so you have a single odor receptor nerve may respond to different odorants, right? So nerve A may respond to eucalyptus, B might respond to banana and almond, and C might respond to all 3, right? So it just depends on the nerve basically in regards to what they respond to. Right, but what's really important for us to hone in basically on is that each neuron only expresses one type of receptor. This is obviously a 2,000-foot view, but let's say, example, a neuron basically is sensitive to almonds, right? It only has let's just say it only has almond receptors on it. It can have 1,000 almond receptors, but it's only going to have almond receptors. It's not going to have receptors of different kinds. Right, so that's important for us to realize, because when we talk about olfaction, it's going to be a lot of label lining or things that are going straight in line one after another that are very single in type. So that makes sense when I talk about them in the upcoming slides. So first thing first is that what happens basically when we smell something? What's the pathway, right, that this odorant takes? Well, we know odor receptors are GPCRs, right? But what pathway does it take, right? So the pathway that it takes, this first route is one of the routes. We actually talk about two, but this first route is kind of similar to a GS route, right? Because we know, GPCR activated, adenylate cyclase produces cyclic AMP, et cetera, et cetera. It's kind of similar to GS, right? But it's actually it's using this olfactory G protein, which is why they call it a G-olf, right? So, but it's very similar to GS in its pathway, right? And we'll discuss the pathway of this upcoming next. So here's the first route. So we know you smell something, basically you smell something new and you're like, okay, how did I realize that smell? How did I smell those almonds from here, right? So reason being is that odorant that's in the nasal cavity activates your GPCR, takes this GS similar pathway in the beginning, so G off is released, cyclic AMP is produced via adenylate cyclase, and this CNGA channel opens. That's basically a cyclic nucleotide gated channel, right? And cyclic AMP is binding to that, right, and causing the influx of calcium and sodium. And we know, remember from earlier, lectures from the last block, influx of calcium and sodium are highly associated with depolarization, right? So that's going to lead to depolarization of the cell. But that's the first part of the pathway, right? So it seems very similar to, obviously, the GS pathway. But here, basically, again, so now we're going to follow calcium because calcium is going to be a very, very important second messenger, right? So calcium then is going to activate this chloride channel, right? So when calcium activates this chloride channel, it causes efflux of chloride, which is very interesting, right? Because we're used to chloride influxing, but effluxing of chloride, since chloride is an anion, that's going to lead to depolarization because negative ions are leaving the cell, thus making the cell more positive, leading to further depolarization. And of course, after this calcium is done, it's going to be going in and out via the exchangers and pumps in order to get calcium back to normal levels, because you don't want to have calcium just floating around like that, right? So again, calcium is very associated with that channel. So you have calcium influx via the CNGA channel, so cyclic nucleotide gated channel, will activate which channel and lead to which event? Give you a few seconds. Okay. Cool. Seems like everybody's on the same page. So remember, sodium and calcium are going to influx via that CNGA channel, right? So, and it will activate, that calcium is going to activate that chloride channel, right? That chloride channel and lead to efflux of chloride because that leads to depolarization. Remember, cyclic AMP is associated with that CNGA channel. So cyclic AMP helps open that CNGA channel, but calcium helps open that chloride channel, right? So those are two important things we're going to want to note. So, but very good job on that one. So we have olfactory adaptation. What that is basically, so when you walk somewhere, or you walk into a new restaurant and you smell something, or you're like, oh, this restaurant smells like, I don't know, some type of food, right? And then you're like, okay, but eventually you stay in there for like a few minutes and that smell goes away. But the next person that walks in is like, hey, this restaurant smells like this, right? So why did you not smell it anymore? The reason being is because you have this olfactory adaptation, right? You reduce that odorant sensitivity so that you're not constantly firing signals about something that you already smell, right? So we have different types of adaptations. We have an initial, we have a short term, which would be a little bit longer, and then long-term adaptation, right? So initial adaptation would be when you basically, that smell goes away very quickly. What's happening is that, remember, this CNGA channel, which is highly associated with cyclic AMP, right, binding and causing the influx, calcium comes in, and binds to this calcium binding protein. This calcium binding protein is actually associated with this CNGA channel and leads to desensitization. Right, so this is just a quick initial adaptation. Calcium comes in, binds to CBP, and leads to that desensitization. That's that initial adaptation. Short-term adaptation, basically what's happening here, calcium calmodulin protein kinase 2. What's happening here, calcium can also bind to this calmodulin right here, protein kinase 2, and then lead to increased desensitization of adenylate cyclase. Right, so short-term adaptation is associated with desensitizing adenylate cyclase. Initial adaptation is associated with desensitizing its own channel, basically that it came out of. Right, so calcium initially came in through that CNGA channel, so initial adaptation is going to be via the CBP. Short-term is via this calcium calmodulin protein kinase 2. And then lastly, we have long-term. So long-term basically is going to be, after a long period of time, calcium can mediate nitric oxide production, eventually to cyclic GMP. And then what happens is that cyclic GMP is going to cause persistent calcium influx. And you're thinking, hey, doesn't calcium influx lead to depolarization? Well, yes, it does, right? But when you lead to too much calcium going inside of the cell, it would actually desensitize the cell, because that's not good, right? You don't want too much calcium going into the cell. So it actually desensitizes the CNGA and adenylate cyclase channels, right? So that's how this long-term adaptation works in regards to, in comparison to the initial and short-term. But again, remember that sodium-calcium exchanger is going to restore that balance after it is all done. So we talked about the first route, which is similar to GS. Right, the second route is similar to GQ, right? And again, it uses this olfactory G protein golf, or G-off, right? He doesn't like golf, G-off. It's going to activate this phospholipase C, right? So again, remember, when you activate phospholipase C, IP3, DAG, etc., we know that that's going to be associated with that GQ route, right? That's GQ. GS is associated with adenylate cyclase, cyclic AMP, right? Those are going to be the main differences between those two pathways. Okay, so if someone walked into a new home and smelled Febreze initially, why would they eventually not smell the Febreze after being in the home for only about a few minutes? This is initial adaptation. Give it a few seconds. Okay. So if someone walked into a new home and smelled Febreze initially, why would they eventually not smell the Febreze after being in the home for only about a few minutes? This is initial adaptation. So remember, initial is going to be calcium binding to that, because it initially comes in through that CNGA channel, calcium binding to calcium binding protein and desensitizing the CNGA channel. Right, A is going to be short-term adaptation, right? So calcium binding to calcium calmodulin protein kinase 2 and desensitizing the adenylate cyclase. And C is going to be long-term adaptation. Remember, it's chloride efflux and not chloride influx. Chloride efflux is what leads to depolarization. Okay, so here
I have a question regarding that. So for short term, what would like the timetable be, or would they just say short term?
Yeah, he's not going to give you timetables in regards. He would just have to say short term.
Okay, got it. Thanks.
Yeah, no problem. That's why I wrote initial adaptation, because I don't want you guys to assume, oh, maybe it could be short term, right? Good question, though. Okay, so we have vomeronasal organ in non-ape mammals. So basically, this is just similar to olfactory epithelium. It's just used for pheromones, right? And does not use golf or G-off. So I would say if you just note what's in purple, that should be suffice in regards to this slide. Okay, so here, again, we have second group of chemosensors. You have trace amine associated receptors. This is just saying that we have different types of receptors that also help with smell, right? It's not only that GS and GQ pathways that are associated with G-ALF. We also have trace amine, basically associated receptors found in humans as well. Uses G-ALF as well, but I think just knowing that it uses G-ALF and it's used for specifically pheromones should be suffice. So glomerulus, so each olfactory receptor, right, is going to express a single, each olfactory receptor neuron expresses a single olfactory receptor. We talked about this early in the lecture. Remember, like, for example, if you're smelling almonds, right, so you would have an olfactory receptor neuron dedicated towards almonds. And that olfactory receptor neuron expresses only almond receptors. Obviously, this is just hypothetically speaking. So, and then it's only going to be those almond receptors, right? So, in each glomerulus is going to receive from only one type of odor or olfactory receptor, right? So that almond receptor is then going to go to a single glomeruli that is going to be dictated or basically used for just almonds, right? So even the glomeruli are specific to a single type. So again, you have, let's say, an olfactory receptor neuron dedicated towards almonds. That olfactory receptor neuron is going to have only almond receptors. Those almond receptors are only going to go to the almond glomeruli. Like, obviously, this is very hypothetically speaking, but that is going to be what's happening here, because you can see these red fibers going to only this glomeruli, purple fibers going to only this glomeruli, blue, etc. Right? So that's very, very important to know. And then eventually from these glomeruli, we'll talk about them, but you'll have your mitral and tufted cells, and then eventually your granular layer. Okay, so here's our olfactory bulb in 5 layers. Right, so you have olfactory receptor nerves. Again, this makes a lot of sense if you think about the pathway, right? So you smell almonds, it comes into your nose, right? And then you're like, okay, what's the first thing that happens? We remember those little cilia in your nasal cavity picking up that odor receptor. It goes to the olfactory receptor nerves, right? So then we know, okay, it goes to the almond olfactory receptor nerves. Those almond olfactory receptor nerves are only going to have almond receptors. Those almond receptors are going to almond-specific glomeruli, right? So that's why that's the next thing, odorant-specific glomeruli. Odorant-specific glomeruli go into that almond-specific glomeruli go to that external plexiform layer, then into that mitral cell layer and granular cell layer, right? So this is going to be the order in which this happens. You're also going to have lateral inhibition in the olfactory bulb, which is very important, right? Because when we talk about the eye, there's going to be, I think it's the next lecture after that, there's a lot of lateral inhibition in order to like see things clearly. Same thing in the olfactory bulb. And this is important because, for example, you want to make sure you're smelling almonds and not smelling urine, right? So you'll inhibit the urine receptors, for example, so that you only smell almond. That's why we have lateral inhibition, to have very, very precise smelling or sight when we talk about vision. And again, you're going to want to know all these 5 layers. You want to know them in order as well. Okay, so olfactory nerve, basically, so this is going to be cranial nerve number 1. So again, remember, this single pattern is this even goes all the way up to the mitral and tufted cells. Remember, mitral and tufted cells only connect to a single glomerulus. So again, everything single, right? So almond coming into the nose binds to an almond receptor, a single almond receptor, or a single almond neuron. That almond receptor neuron is going to have almond receptors. Those almond receptors only go to almond glomeruli. And those almond glomeruli are only going to connect to basically one mitral and tufted cell, right? So mitral cells connect to single glomerulus, right? So you can see vice versa, that's what I meant. Yeah, so mitral and tufted cells, they're going to be binding to a single glomerulus. So you can see here's this mitral cell binding to a single glomerulus, this mitral cell, etc., etc. So mitral cell pathways. So what's going on here is that mitral cells are going to have more pathways than tufted cells, right? I think tufted cells have around like 2 to 3 regions they go to, but mitral cells are going to be 5. So there's a few pathways that goes on here, but of course the one that's very important is the one indicated in red. But the first one we talk about is this thalamus going to this orbital frontal cortex, right? That's important for odor perception and discrimination. So basically, what are we smelling? Are we smelling almonds? Are we smelling urine? That's what that pathway is important for, so we can see that indicated here. We also have a very important pathway, which is going to be an odor-induced fear signaling, basically, via this amygdala and piriform boundary. Right, so that's very important because remember, we know odor is very tied to like memories and stuff like that. So you can smell something and start remembering childhood memories. It's the same thing with fear, right? You can smell something and be extremely fearful of it, right? So if there's a food that you extremely hate, right, you smell that food, you may even be fearful of it, right? That's an example of this odor-induced fear signaling, right? And that's via that amygdala piriform boundary. That's what you're going to want to know. Okay, so to no surprise, remember that this olfactory pathway has labeled line approach or labeled line coding. You can see everything is single, basically lined. Remember what we just talked about, one line here, eventually a line here, and it doesn't mix until it gets into this olfactory cortex, which makes sense because then we're going to need to discriminate between smells, right? And in summary, basically, unique odors basically are going to activate unique patterns of odor receptors. So depending on the odor receptor, it may be, responding to eucalyptus, or it may be responding to banana and almonds, etc., etc. So here's olfaction as a diagnostic tool. Obviously, it's very sad, right? At the age of 36, I feel like super young, right? So you typically start having a loss of smelling pleasant odors. Obviously not good. And these are some of the diseases that are associated with impacts of olfaction, right? So IPD, idiopathic Parkinson's disease, tend to be anosmic, which is basically loss of sense of smell. Huntington's has a little loss. Alzheimer's is more so associated with a central loss, so like with the central pathways. COVID is going to be a decrease in olfactory sensation. So obviously he put this slide in here this year, right? He didn't have this slide in there last year. But the thing to take away, I would say, from this slide is going to be that COVID can, affect us, likely affects the tentacular and the horizontal basal cells, right? I would say that's the most highest thing to know here, just because, there's a lot of unknowns in regards to COVID and especially about olfaction with COVID. So nothing too testable here. Only thing I would note would be what is in red. Okay, so what is the order for the olfactory bulb layers from distal to proximal? Remember, distal to proximal. I'll give you a little bit. I know this is a little tough. I'll give you a few more seconds. There's a lot of answer choices to look at. Okay, so what is the order for the olfactory bulb layers from distal to proximal? Right. So again, you just have to go through your pathway in your head, basically, in order to obviously conceptualize it to make it much easier. Right. So again, we know, almonds coming into the smell of almonds coming into your nose. You're going to activate those olfactory receptor neurons. Remember, that's specific to almond. Those almond receptor neurons only have almond receptors. Those almond receptors go to almond-specific glomeruli, right? So we can already knock off B and C. And then we know almond-specific glomeruli are going to that external plexiform layer. Then it's the mitral cell layer, and then the granule layer. The way you can remember is maybe the granule layer is the grand finale, right? So it's the last one. Right, that can probably help you remember that granule layer is after mitral cell layer. right? So that's going to be the order. Very good job with that one. I see that a few of you have definitely put that answer A, so really good job. I know it's new information. Okay, so here we have peripheral and central organization of the gustatory system, right? So we talked about cranial nerves a lot in anatomy, right? So yeah, that was awesome. But here we're also going to need to know it as well in regards to taste. Right, so we know the facial nerve. Remember when we talked about this in anatomy? Anterior 2/3 of the tongue is getting facial nerve, specifically chorda tympani, and that's via that geniculate ganglion. Right, glossopharyngeal is going to get the posterior 1/3 of the tongue, so that's going to be via the petrosal ganglion, and then vagus nerve is going to be remember that's more so associated with the pharynx, and that's via the nodose ganglion. Right, so not only do you want to know the nerves, but you also want to know the ganglion associated with them. I remember that being important as well. So not just the nerves, but you also want to know the ganglion that are associated with them. And here are just some taste cell locations. Okay, so we have taste buds basically embedded in papillae. Again, this is what I just talked about: the anterior 2/3, the posterior 1/3, and the pharynx. Right, but a little bit about the taste buds is that you have fungiform and filiform taste buds in the anterior 2/3. We don't have a picture of filiform; they're very, very small. But fungiform we do, which is going to be on the bottom right here. And you also have basically in the posterior 1/3 foliate and circumvallate, which is going to be very, very large. You can actually see these if you went into a mirror and just put out your tongue. You can kind of see those circumvallate basically taste buds. They're very large, right? And that's associated with the posterior 1/3. We have roughly 4,000 taste buds on the human tongue, found at the base, contain numerous taste cells. But what's important, right, is that taste cells turn over about every 10 days, right? So if you're ever burn your tongue on hot pizza, something like that, don't worry, your taste cells are going to turn over, right? So, but don't burn it obviously all the time. But, the taste cells are going to be replaceable very, very quickly, which is great, right? So here, we have tastings in different thresholds, right? So the reason why I bolded bitter and sweet is that bitter has the lowest threshold and sweet has the highest threshold. And evolutionarily, that makes sense, right? Because something that's bitter is actually, can be literally like more dangerous, right? So for example, things that are bitter, can be poisonous, they can be dangerous, right? Which is why we have a very, very low threshold for that in order, for like survival and stuff like that. Sweet, obviously things taste good and stuff like that, but that's not, obviously necessary for survival, which is why the threshold is much, much higher, right? So that's why bitter, it has a super low threshold so that we can tell when things are bitter. And again, contrary to public perception, taste is different types of tastes are going to be all over the tongue. You don't have a side of the tongue that's dictated to sweet, a side of the tongue dictated to sour, et cetera, et cetera. But to this day, if you Google that stuff, it's still online. So you can Google it if you wanted to, and it'll still be there. But don't believe it. Okay, so we have types of taste cells, right? So we organize this. So I organize this basically into 3 different types. You have type 1, type 2, and type 3. So with type 1, basically what's important is that it's kind of like a salt sensor. I mean, it's associated with being a salt sensor, but they're not 100% sure, meaning he most likely won't test you on type 1. Right, so it has support function, might be a salt sensor, but most likely not going to test on that. What's important are these type 2 and type 3s, right? So again, type 2 is going to be bitter, sweet, and umami, right? So umami is basically like that meaty or savory taste, and it's going to be when they release basically their neurotransmitters, it's not going to be conventional. It's going to be via pore release. That's really important. Right, so type 2 cells, what happens is that they release ATP, for example, just out of pores. They don't use how we talked about vesicle transport and vesicles leaving or releasing a neurotransmitter, et cetera, that typical pathway. Type 2 cells don't do that. They do non-conventional, basically, synapses, and they just release ATP via a pore. Right, and then we have type 3 cells, which are associated with sour, and those do typical synapses, right? They vesicular release of ATP and serotonin. And lastly, we have our basal cells, which is just our progenitor cells. So this right here is just indicating how many neurons are, how numerous the neurons are. Type 1, we have more type 1 cells than we do type 2, and more type 3 cells than we do type 1. It's roughly around 90% type 1, 9% type 2, and 1% type 3, which is really interesting, right? Because we don't seem to know too much about type 1, but that's 90% of our taste cells, right? So I don't know. I find that quite interesting. And type 2 is 9%, and then type 3 is going to be roughly around 1%. Okay, so I broke this up basically into two different slides in regards to the chart. So we have mammalian taste cell receptors in cells, right? So you have umami, sweet, and bitter. These are all going to be GPCRs, right? So type 2 cells, I would get into your head that type 2 cells are GPCRs, right? So umami and sweet are going to be heteromeric, right? So they have two types of GPCRs. You can see that umami is going to be non-specific as well as sweet. And what that means is that umami basically receptors can respond to all different types of savory and meaty tastes. Right, sweet, the same thing. It basically, this receptor can respond to a ton of things that are sweet. Right, it's also important to note the specific receptors. Right, you want to know that umami is T1R1, T1R3, and that sweet is T1R2, T1R3. Right, so I would know that. Bitter is going to be monomeric, right? So they're not sure it's monomeric, obviously, but because of a question mark, but they have some research that says that it's monomeric. Right, so, and these are going to be our T2Rs, right? So, and they're high affinity. So, in regards to bitter receptors, you have a specific subtype in regards to different types of bitter taste. So, it's not, it's very specific, right? It's not non-specific like how umami and sweet are. Bitter is going to be very specific in regards to the bitterness that it responds to, right? So, that's going to be a major difference between bitter and umami and sweet. But again, remember that these are going to be associated with type 2, right? So type 2 cells. Okay. Oh, and one more very, very important note is that type 2 cells basically are only going to express one receptor at a time. And what I mean by that is that, for example, one type 2 cell will display all umami, and another type 2 cell will basically display all sweet. And another type 2 cell will display all bitter, right? They're not going to have all three within the same cell. That's very important because they actually do some research on that, and I'll show you in the next upcoming slides of how they proved that there's only one type of receptor per cell. Okay, so mammalian taste cells and receptors. So you have ENaC and sour. These are the other two receptors. Important to note, these are ion channels, right? So type 3 cells are ion channels. Type 2 cells are going to be GPCRs, right? So important for us to differentiate those two. ENaC, basically, I mean, they're unsure about the cell type and possibly type 1. What they do know about ENaC is that it responds to sodium at low concentrations. Sour, on the other hand, is going to be type 3, complex channel. But remember, this PKD2L1 is going to be associated with sour. And again, what's very important about sour is that it's based on intracellular acidification. And what I mean by that is, so if someone was to, drink HCl, obviously don't do that. HCl is extremely acidic, right? But it's not going to give you that sour taste in your mouth, right? It's not going to taste like a Sour Patch Kids, right? So it's probably going to burn your mouth or something. So reason being is that these sour receptors are responsive to intracellular acidification. So things that are more permeable to that sour receptor or sour membrane are going to be considered more sour, right? So it's that intracellular acidification, not that extracellular acidification that is associated with sourness, right? So that concept is important to understand. So type 2 cells, again, remember, these are released by pores. They are not vesicles. Remember, our type 2 cells are going to be our bitter, umami, and sweet. So if you want to remember that, BUS, right? So BUS, bitter, umami, sweet. Those are going to be for your type 2 cells, right? So no classic synapse. And again, G-gasteucin is actually going to be similar to GQ, which makes sense because you can see this G-gasteucin actually activates this phospholipase C, eventually IP3 and DAG. and eventually, after all activating all these channels, leads to the release of ATP. Right, and I find this quite fascinating because ATP is a pretty large molecule in respect to these other neurotransmitters, and basically this membrane has to widen pretty far in order to let ATP come out of the cell, and this is going to be basically acting as a neurotransmitter, right? So quite interesting how this type 2 is going to signal transduction, but important to note that G-gasteusin is associated with type 2, right? So G-gasteusin, this pathway is associated with type 2. Okay, so here, what's happening is that you have ENaC cells as well as bitter receptor cells, or bitters. Remember, bitter receptors are part of that type 2. Remember, type 2, we have bitter, umami, and sweet, right? So what's going on here is that if you were to, you're eating dinner and you can tell when something's a little salty, right? So you're like, oh, this is a little salty. How do you detect that? That's going to be via ENaC, right? ENaC is very important for low salt concentrations, right? So it's only sensitive to NaCl, right? It's like that table salt. So low salt concentrations is via ENaC. But if you were to just, taste something that was just so salty to the point that it was just like, at that point it was just nasty, it was just like bitter, you're not activating those ENaC channels anymore. You're activating the bitter receptors because it's so much salt, it's just bitter at that point. So the bitter receptors get activated at very high salt concentrations, also known as low salt sensitivity, which leads to salt aversion, right? So it makes sense that ENaC is going to be activated at low salt concentrations, but the bitter receptors are going to be activated at much higher concentrations, right? And that's shown in this study right here. You can see in the control, you can see via this black line, when you increase salt concentration, you increase the firing rate is going to continue. But when you have amiloride, which inhibits ENaC, meaning that at low concentrations, nothing fires. When we're looking at the blue one, then it doesn't fire until you get around 250 millimoles of NaCl, which shows that, okay, we know amiloride inhibits the ENaC channels. Thus, no ENaC channels should react at low concentrations. But when we get to super high concentrations, even though ENaC is knocked out, our bitter receptors come in and respond to all this salt, basically, because it's way too much. Basically, it's high concentrations of salt, right? So you can see the differences between those two. Here, what we're proving here basically is that diphtheria is killing specific cells. So here, it's killing the sweet receptors, and this one is killing the sour. And what this proves is that each receptor is going, or each cell is going to have one single receptor. Remember, like we said, type 2 receptors, there's bitter receptors, there's sweet, and there's umami. If you were to knock out the sweet, the umami and bitter still work. And the only reason that can make sense is that each cell represents only one receptor, right? Because if each cell represents multiple receptors, the cell would die, right? Because it had all these receptors, knocked out sweet and knocked out the rest of them. So what this is saying is that basically only one receptor is going to be associated with a specific type 2 cell. Same thing with sour in regards to type 3. So here, PKD, remember when we talked about the PKD being very important for sour? Yes, it's important for sour, right? But if you were to do, unfortunately, I know this could have been groundbreaking research, but unfortunately, PKD knockouts, right? You reduce sour sensing, but it's not eliminated, right? You can still taste sour even when PKD is eliminated, which means that PKD, has a function basically in sourness, but it's not vital basically for sour sensing. So it must be another mechanism that we don't know about. Here, I just put this slide just because he talked about the alternative mechanisms of acid and sour taste transduction, but he's not going to test on this because none of this is sure. So I'm pretty sure you can X this out. All of this is just in the works in regards to research. Okay, so if someone dared you to eat a pound of salt, which taste cell receptor will most likely be activated? Give you a few seconds. Any questions? Is everybody good? Turn this light on. Okay. So if someone dared you to eat a pound of salt, which taste cell receptor will most likely be activated? We know this is going to be, okay, at low salt concentrations, we activate ENaC, but at high salt concentrations, it gets to the point where it's super bitter. So it's activating the bitter receptors, which are going to be our T2Rs, right? Remember that our T1R1, T1R3 is going to be umami. T2R2 and T1R3 are going to be your sweet. And ENaC is low concentrations. Okay, so we have communication between the taste cells and the afferent nerves, right? He didn't have this slide in here last year, which makes me believe that this is important for the exam. So I would note this, what it's talking about. So we have intragemal fibers, which are basically going to be going within the taste bud. We also have perigemal fibers, which are going around the taste bud, right? And what's happening here is that the intragemal fibers are going to be important for gustation, which makes sense, right? Because they're going within the taste bud. So that's going to be associated with, we should know these cranial nerves. We know cranial, that's a facial nerve, glossopharyngeal and vagus. The perigemal, since they're wrapping around the taste bud, but they're not going inside of it, they're going to be important for somatosensory, right? So they're important for kinesthesis. Right, we'll talk about that at the end of the lecture on the last slide, but this is going to be important for kinesthesis. So here, important to note, remember, G-gusteucin is important for the type 2 cell transduction, and P2X2/3 is extremely important for all tastes. So we'll talk about this, but remember that P2X2/3 is critical for tasting anything. If you were to knock out P2X23, you won't be able to taste anything in regards to anything. So it's important for everything, and that's going to be very, very important to know. So here, no obvious innervation of type 1. Type 2, again, remember, that's our ATP pore release. So it's going to be non-conventional. And in type 3 is going to be our conventional neurotransmitter release. And this is the knockout we just talked about. So they did a double knockout of, a double knockout here of P2X2. And they noticed that here they responded to all these different types of tastes. Here, they didn't respond to anything. So basically saying that if you knock out P2X2, you can't taste anything, right? So that's going to be critical for all tastes. That's very important, right? So, and 5-HT3 has a minor contribution. In this study, we know things that we like are going to be things we attract ourselves to, right? So some animals, they like saccharin and sucrose, but they don't like quinine and denatonium because they're bitter. But if you were to knock out this P2X2 receptor, which is purinergic, what's going to happen is that these rats, or basically our animals, are not going to have any preference to bitter or sweet. Again, further proving that this taste, or P2X2, is critical for all tastes. And what that is basically a purinergic receptor. Remember, purines are kind of like, like ATP and stuff like that. Okay, so communication between the taste cell and the afferent nerve. So again, you can see you have your type 1, type 2, and type 3. Important to note is that our type 2 cells actually can activate our type 3 cells, right? So remember, type 2 cells go through that unconventional release of neurotransmitter, which is going to be ATP, via that pore release, right? This ATP can activate this type 3 receptor, right? So that's very important. We also have this thing called NTP dase 2, which is actually going to break down ATP, right? So when ATP is released, it's obviously going to break it down and end transmission, right? So these are going to be the important things we want to know in regards to type 2, type 1, type 2, and type 3. And again, you can see here, even in this picture, it shows vesicular release right here in type 3 cells, which is conventional. Type 2 is just going to have this ATP release channel via pore, which is very, very non-conventional. Here, each taste cell we know is going to be innervated by gustatory fibers, which innervates taste buds. So thus, if you find electrical activity or you stimulate electrical activity, you'll represent the input of many different types of taste, like sugar, salt, acid, or sour, etc. So here, basically, we have the different types of taste cells. And what's important here is that understanding the difference between combinatorial and specialist, right? So specialists basically are going to be cells that are going to be labeled line. Basically, they transmit the same signal all the way up to higher order centers. So in this image right here, you see sweet, for example, goes all the way basically to the higher order neurons and is basically stays as sweet. It's a specialist. Sour, on the other hand, is a type 3 cell. And if you remember from the last slide, remember, type 2 cells release ATP via those pores, right? And that ATP can actually activate type 3 cells. So type sweet, for example, can activate sour. So, for example, like that sweet sour taste. That's why type 3 cells are generalists, right? Because they can get input from multiple cells. They're not specialists in what they do. They get inputs from multiple other cells, which is why they're a generalist, also known as combinatorial, because they're getting a combination of signals from other cells, right? So that's very, very important for us to know. Here, only thing I would know is heterogeneity is found across the entire tongue in regards to taste and stuff like that. So he talks a little bit about the pathways, right? I guess he goes rather quickly through them, but what I would know is what's in this blue box, right? So he talks about, primary afferents, going into the nucleus of solitary tract, thalamus, eventually parabrachial nucleus. But again, we process it and it goes into our conscious perception, right? So this is basically a little pathway of, obviously it takes multiple pathways once it gets into the brain, but there's little topographic organization in regards to these pathways specifically, right? But there's one exception in which we talk about in the central nervous system, which is here. Here, basically, you have overlap between multiple receptors. So what I mean by that is that sweet and sour overlap. No, sweet, sour, and salt can all overlap, but bitter and sweet do not, which makes sense, right? Something can be sweet and can also be salty, right? Something can be salty and also be sour, right? But nothing is going to be bitter and sweet at the same time, right? That doesn't make any sense, right? So those two actually don't overlap, right? And there's actually 3 different types of information we can extract when we taste something. That's the quality of it, the intensity of it, and hedonic value, which is basically, do we like it? Do we not like it? If you don't like it, you'll have basically a protective behavior, which can be, okay, avoid that at all costs. If you like it, you'll eat it more, for example, et cetera, right? And then lastly, we have our chemesthesis, which is basically going to be sensation. Remember, chemesthesis is not the same thing as gustation. This is going to be sensory. So for example, this is regulated by the trigeminal nerve, and it's going to use some nociceptor subtypes. So for example, I would 100% know these 3. I do remember this being important for the exam because this is the only slide on chemesthesis. TRPV1 is important for chili, heat, and acid. TRPA1 is wasabi, and TRPM8 is going to be menthol. And the way I remember that is M for menthol, and menthol we know is like, in mints and stuff, right? So that's associated with like cold, coldness in the mints and stuff. TRPA1 is wasabi, and then TRPV1 are going to be these 3, right? And that is all. Anybody have any questions?
Yeah. So back for the ORNs and the glomerulus, I didn't quite understand how the ORN synapses with the glomerulus at first, because he said something about each olfactory receptor going to 2 glomeruli, but then we kept talking about 1-to-1 because he was saying like we only have 1,000 of the receptors, but we have 2,000 glomeruli, and I was just wondering if I just like misunderstood him.
Yeah, I mean, you can have, 2 glomeruli. This is obviously 2,000 foot view, but 2 glomeruli for almonds, right? So it just depends, but those are going to be specific to one specific olfactory receptor neuron. Does that make sense?
Yeah, so like each olfactory receptor is only going to do, like, say, almond.
Yes.
And then, but they can go to 2 different almond glomeruli.
Yeah, exactly. Yeah.
Okay.
Obviously,
Okay.
super hypothetically speaking, but yes.
Okay. Cool. I just want to make sure.
Yeah, got you. All right. Any other questions?
I think I probably missed this, but on slide 40, you had the last bullet crossed out. Maybe I printed my slides like earlier than like maybe he tweaked it or something, but we don't have to know that. Like, did he cross that out or was
Yeah,
that?
I'm pretty sure he crossed that out, right? Yeah, because I downloaded it and he crossed it out.
Oh, okay. Thank you.
Yeah, no problem. Okay. Any other questions? Yeah, so yeah, no, great question, Daniel. Let me pull that up. So I'll show you because I think showing with the picture will be easier to tell you. So right here. Okay. So okay, I think you see my screen. So basically, so frequency is what's important in regards to if something is in phase, right? So obviously, things that have the same frequency have the possibility of being in phase. Of course, if something starts at the same time, right, and has the same frequency, it will be in phase. But you do not have to start at the same time in order to be in the same phase. Does that make sense? Reason being is because, for example, this frequency we know is the same as this one, right? If this basically started right here, it would be in phase because the waves would match exactly as basically as they say. Obviously, it doesn't start at the same point, but when it starts, it becomes in phase with that sound, right? Regardless if it starts on this dot, or eventually on this dot. Oh, remember this one. Does that make sense, Daniel? Okay. No, it's a really good question. Yeah. But yeah, obviously, if they start at the same time, that would obviously help a lot and make sense as to why that would be in phase, but not a requirement. Okay. Any other questions? Okay. If not, good luck with your studies. Let me know if you need anything and enjoy your Friday and your weekend, okay? Take it easy, everybody.